I want to talk about, what I want to talk about is our kind of efforts in the technology development space where we, like you introduced, we developed all of the single cell technologies for histone modification profiling and deciphering the epigenome, and then also the way how we apply this to study the complexity of the brain. So the big question that my lab and we are interested in is the transcriptomic diversity and kind of the cell type diversity of the human brain. So the transcriptomic studies we know from these that human brain is especially complex. So every individual has more than 80 billion neurons just in the cortex, and um, yeah, they're incredibly diverse, many different cell types that emerge during the development. So using the current single cell approaches, we are basically just scratching the surface. The largest atlases so far have been of millions of cells, so we are several orders of magnitudes lower than actually the complexity of each individual. But from what we have already collected, we know that there's thousands of the different cell types that present, and the central question that my lab is interested in is how is this intricate architecture programmed and controlled at the gene regulatory level. So gene regulation is really driven by chromatin. It's driven at multiple different levels, but one of them is really the chromatin. So the process of activation of transcription through the RNA polymerase involves a recruitment of transcription factors, activators, co-activators, and repressors to various cis-regulatory elements, either proximal to the promoter, so here when transcription start site is, or distal at the activators, repressors, or enhancers, and so on. So really interested in studying the different chromatin states that drive this process of gene activation and gene repression. So the current, so the cis-regulatory elements, to dive a little bit deeper into the introduction, there can be many different kinds. And the current really state of the art and kind of classic technology in the field to study this is to look at the chromatin accessibility using single cell ataxic. And typically we'll find that most of the cis-regulatory elements, they would have some level of and chromatin accessibility that's driven by transcription factor binding, and then chromatin remodelers being recruited and opening up the chromatin so that it's in this permissive open state. So what I would argue is there's multiple layers of regulation and multiple layers of the chromatin modifications that are also there, but we are currently, or have been for a long time, kind of blind to it at the single cell level. So looking at HTK4 trimethylation on the promoters or monomethylation on enhancers, 20K27 acetylation on the enhancers, and these are all the modifications of the histone tails, basically, on the nucleosome. So these can mark either active promoters, if you have a specific combination of K4 trimethylation and K27 acetylation. We can also find poised or primed enhancers, which are only labeled by K4 mono, poised promoters, which have K4 tri, but no K27 acetylation, active enhancers, and so on and so on. So multiple different states that actually all of them would appear accessible if you only see chromatin accessibility data, but then the histone modification really add more and more layers, and this combinatorial code of histones can be used to predict functions of these cis-regulatory elements. We have also other modalities like demethylation tends to be depleted and at open chromatin regions, but also CTCF, which defines the chromatin architecture and also serves as an insulator module that insulates cis-regulatory elements from each other. And of course, the chromatin architecture, so 3D organization of the chromatin, the looping of these cis-regulatory elements. So we really need integrative approach to study this chromatin structure. And that's what my lab has been trying to, and what I've been doing in my postdoc, and now my lab is trying to push for. So what we want to do is use histone mark as a proxy for enhance and promoter states or activation states or repression states. We want to look at the repressive histone modifications. I'm not showing here, but we also have more layers of repressive histone modifications such as K27 trimethylation or K9 trimethylation. And then also we want to resolve complex chromatin states that are defined by combinations of histone modifications like poised or bivalent chromatin. So chromatin is incredibly dynamic. So I just want to show you one example of a study that has been recently finished at KI, in the lab of Stan Linnarson, when they did both single-cell RNA sequencing and single-cell ATAC-seq in developing human brain. I would actually find that on the transcriptomic level, you find hundreds up to thousands of different genes being variable. So here's 1,300 genes across 130 clusters that defined in this study. But on the level of the chromatin, you actually have much more variability. So to define the same 135 clusters, the multi-ohm data, you can actually use, you can find more than 100,000 variable cis-regot elements. So the gene expression um, um, is really, there's much less variability in terms of gene expression and the cis-regot elements. There's much more space for combinatorics yeah, they are more specific to cell clusters than the individual genes. So what I want to introduce before I start diving deep into the technologies that we developed is a bit of the principle of this TN5 transposition, which is really central to all the methods that we have been developing. And this has been originally developed by Jason Buenrostro in the lab of Will Greenleaf at a single cell level. So what they found is that if they put TN5 transposase, this is a recombinantly purified bacterial enzyme, but if you mix it with nuclei, a mammalian or any other nuclei, the transposase will specifically only attach to the open chromatin and introduce linkers that is preloaded with into these open chromatin regions. So there is no so-called transposition or tagmentation in the closed chromatin, but all of this specifically happens into open chromatin regions. You need two transpositions events close to each other, and then you can basically just use the red and green parts to amplify these regions from the chromatin and sequence specifically only the open regions. So we have been using this kind of similar approach in technologies that come under the CUT&Tag family. So this has been developed originally in the lab of Steve Henikoff, but we have been using extensively and pushing the boundaries of the technologies further. But in CUT&Tag, basically what you do, you can use antibodies targeting histone modifications. They're very specific antibodies that have been used for decades in ChIP-seq experiments. And you can actually bind them to the nuclei directly, fresh nuclei, not fixed, and then recruit a TN5 transposase to these antibodies through a protein A binding domain. And this specifically tagments now not open or closed chromatin, but specifically regions that are recognized by the antibodies. And this works actually for both open and closed chromatin, so you can target both active histone marks, tend to be more in the open parts of chromatin, but also repressive histone marks that are more in the closed chromatin. So this is the key principle that we are using in most of our technologies to recruit TN5 to different chromatin features. So combining this CUT&Tag protocol, binding specific antibodies, targeting histone modifications, then we use also secondary antibodies to amplify this signal, and then protein A transposase, activating the transposase with magnesium. You can perform these CUT&Tag experiments. So one of the first, when I was in the lab in around 2020, 2021, was the first thing we asked is whether this could also work at a single cell level, combining with the 10x chromium single cell ATAC-seq, because the transposase chemistry is basically the same like you would use in ATAC-seq experiments. Is the recruitment which is different. So we used kind of the standard 10x genomics protocols, the 10x genomics chromium single cell ATAC, and combined with the antibody targeted tagmentation. And this has worked. So we tested it first on a mixture of cell lines. We use a mixture of mouse embryonic stem cells, fibroblasts, and neuronal lineage. We performed this antibody directed tagmentation, optimize the protocol so that we can use it for the chromium single cell ATAC, analyze the data, and what we found is that we can actually, in the original mixture of these three cell lines, we can identify three clusters now based on the histone modification profiles. So we use antibodies targeting HTK27 acetylation in this case. Sorry, HTK27 trimethylation. So when you benchmark against the publicly available data, so here's the single cell CUT&Tag data now after clustering and pseudo bulking. We can find the profiles of embryonic stem cells correspond very closely to the ENCODE data that was previously generated in ChIP-seq. For the treated fibroblasts, also corresponding nicely with the ground truth data and then also for the only new neuronal cell line, it also corresponds very well. So this seemed to work really well in the cell lines, but can we actually translate this to a tissue where it's much more interesting? The heterogeneity that we are interested comes from the tissue. So we apply this to mouse brain at the developmental stages of postnatal A15 to 25, dissociated the brain and extracted the nuclei. We performed this antibody-direct augmentation again. There were several different histone modifications and the TEDx Chromium single-cell ATAC protocol. So this is the data that we obtained. So long story short, basically this works really well. After profiling a number of samples, we could specifically identify all the different major cell types in the brain based on H3K4 trimethylation profiles. So this marks promoters. So we can see the active promoters define the cell types. H3K27 trimethylation, which is a repressive histone modification, which suppress is a polycomb mediated modification that suppresses most likely alternative sulfates or progenitor genes as well, as those important functions in development. H3K27 acetylation, looking at enhancers and promoters, and also H3K36 trimethylation, which is a mark of elongating polymerase. So the different histomarks have a little bit different powers. So some could resolve a little bit worse the cell types. Some worked really well, very efficient. But I think what this shows is that this can be really widely applicable to different types of histone modifications. And it's one of the first methods that has been, there has been single cell chip-seq technologies developed before, but they have not seen much adoption because you need specialized equipment and really complex protocols take a long time to set up. So it's a relatively simple protocol that can be performed in a day, and you can obtain this kind of data very quickly and easily. So looking at the markers, here I'm plotting pseudo-bulked profiles. So for each cell type that we found for H3K4 trimethylation, we look at the promoters of the marker genes. So for astrocytes, RFX4s need to have a lot of K4 trimethylation. For oligodendrocytes, MOC, and these are well-established markers. So this really works well and can give specific promoter chromatin data in these cell types. So this, I think this also nicely summarizes the power of the technology, where you can, in kind of a few experiments targeting a few different histone modifications, identify really the heterogeneity of the chromatin states, looking at now at all these four marks, K4, K27, methylation, acetylation, and K36. We can really see that the K36 is enriched. This is a gene expressed in oligodendrocytes. This mark is enriched in the gene bodies. There's a number of enhancers in the introns of this gene. The promoter is heavily K4 trimethylated, an active state in oligodendrocytes. And we see some gene repression of this gene that's neighboring this marker gene. So rather than doing all the laborious sorting, purifying a population, doing bulk chip-seq, you can get this kind of data very much more efficiently in the heterogeneous samples. So this has the power of the single cell technologies. So we really try to share this kind of technology. We published a protocol on protocols.io, try to help as much as possible to people to adopt this technology. And I think what is really satisfying and nice to me is that there's a button in the protocols.io that you can press if the protocol has worked, and we have a lot of very good feedback from this. We also have a lot of comments, and you can see 96 comments. This may be a little bit old screenshot also, I don't know, maybe it's more. So encourage the people to comment and ask questions so that also the answers are public and everybody can see them, and it can help them to establish the technology. So this single cell cut-and-tag has worked well to profile individual histone marks, but then we asked the next question is whether we can develop kind of a similar approach to this, tethering TM5 transposase to an antibody, but on a multiplexed level. And that's how we developed a new technology called nanobody-based cut-and-tag. What we do is replace the protein A, TM5, with the constructs of TM5 fused to nanobodies. These nanobodies are secondary nanobodies targeting either primary antibodies raised in mouse or in rabbit. So we now can use two channels, and the TM5 is it's a very nice enzyme because you can easily barcode it. So we can couple the species-specific antibodies to specific barcodes. In this case, barcode A, barcode B, And then on top of that, we can actually do also ataxic with another barcode in the same sample. So in this way, this nanobody-based cut-and-tag allows to do look at the open chromatin and do histone modification at the same time. And just for explanation, what are nanobodies? If you have not heard about these are just very, very small antibodies. So the classical antibody has a too heavy, too light chains. And then the nanobody is really only the variable part has only the variable part and is very, very small. And it can be efficiently expressed in E. coli. So we can easily take this nanobody and fuse it with TM5 and express it in E. coli. And that's what we did. So this allows to look at 3 channels. This is the first bulk data that we got from this profiling. We took a cell line, first performed ataxic with a specific barcode, and then we, after ataxic, you would normally lyse the cells and extract DNA and do PCR in bulk or do single cell indexing. But what we do is wash the cells, or the nuclei, and the nuclei are still intact and can be used downstream. And what we do is incubate these nuclei together with antibodies, raised into different species, and then the nanobodies that are targeting these primary antibodies with different barcodes. And we did, first experiment we did was in bulk. So we have a ground truth data individually profiled, K27 trimethylation, acetylation, and ataxic. And then we have the multimodal measurement where we use this workflow to actually profile all these 3 at the same time in a single experiment. And after demultiplexing and splitting the modalities, we can actually really nicely reproduce the data obtained in a single experiment. So this was nice proof of concept that this kind of multiplexing can work and will work. So we then applied it again to the mouse brain at post-conception day 21, focusing on oligodendrocytes. We dissociated the brain and then performed ataxic and 2 nanobodies, nano-CUT&Tag, together with 10x chromium. And this is the UMAPs we obtained. So now we get from single experiments 3 UMAPs for each of the modalities with all the cell diversity. Hopefully you can also notice that we find, rather than finding just the 8 or 9 main cell types in the brain, we can also find much more heterogeneity in all of the modalities. So we found more than 20 clusters, and this is really zoomed out at the level of the whole brain. I think if we focus on specific brain regions, we'll also find probably more diversity when we also sample more. So this is driven by the high number of unique fragments per cell that also came from some other improvements of the protocol. I'm not going to go into much detail, but what we actually see, what we actually achieve in this protocol is around 10 times higher complexity of the data and sensitivity of the method, which helps to resolve these cell types. So this is the comparison with the first version with the single cell cut-and-tag protocol. And on top of that, we also know the links because it's multimodal data. We know the single cell barcodes across the modalities actually match each other. So we can actually link all the individual modalities together. And we can use all the modalities for the clustering and dimensional reduction for cell type identification, but also we can start looking at the relationship between the modalities, for example, during development. And that's what we did. So we subset this data. Most of these cell types are actually pretty much terminally differentiated, but there is one developmental process going on where the OPCs or oligodendrocyte progenitors differentiate towards mature oligodendrocytes. So we subset for these cells and we perform pseudotime analysis to order them in a pseudotime. And then we actually we can look at all the individual modalities on its own and in relation to each other. We can actually find interesting patterns in K27 trimethylation deposition, these kind of 2 waves. So the first wave of the trimethylation coming already very early when the OPCs are born, and then the second wave coming when the OPCs start differentiating towards the oligodendrocyte fate. We can also correlate this with gene expression using integration techniques, and we can define variable genes that are variable on the chromatin level. Most likely, we thought these genes should also be variable on the gene expression level, and they actually are. So what we see on this heat map is a subset of genes that are variable on the chromatin, and then this is their gene expression levels. The ordering is in the pseudotime as the cells differentiate, as I saw before, the same pseudotime as here. And then we can actually see that actually a lot of the genes are variable, so they are expressed at different phases of this lineage differentiation. For example, either in the progenitors or transiently expressed or late expressed. One of the interesting things that we should find is these variable genes at the chromatin level. They tend to be quite lowly expressed. So this would be not your typical genes that you will find in a single-cell RNA sequence experiments. Of course, single-cell RNA sequencing is looking only in the highest expressed genes. That's where you also have the largest statistical power to define them, right, and identify them. So all the single-cell RNA-seq markers tend to be very, very highly expressed. But our genes, when we look at the defined based on the chromatin, when we look at the gene expression, they're actually several orders of magnitude lower expressed. So this, I think there's also a lot of potential in discovery on kind of the non-classical regulators of the differentiation that are often or can be often overlooked by single-cell RNA sequencing. So we again published these protocols, try to help as much as possible. This was actually the first project in my lab where we wrote down like a very nice version of this protocol. And Jose, the first PhD student in my lab, put together these nature protocols where we share. We also have put out in the same way like with the single-cell cut-and-tag protocols IO version. So this is out there with really a lot of helpful tips on how to establish the protocols from the production of the nanobodies, TM5 sample considerations, and then the incubation, single cell barcoding, and data analysis. So if you're interested in this, check out the nature protocols and the protocols IO. One of the things we are currently working on, now I'm switching gears a little bit to the more current work that we are doing in the lab. So one of the goals of my lab is to even more expand the capabilities of this kind of technology. So we really love working with TN5. It allows a lot of flexible recruitment and a lot of really interesting experiments. So one of the things we came up with together with Letian when he joined my lab as a first postdoc is to design these new fusion proteins now between methylation binding domains. So these methylation binding domains are targeted towards DNA methylation. So now we are not looking at modifications of histones, but modifications of DNA itself, with the idea that these methylation binding domains could be used to recruit TM5 to the chromatin in the same way like use antibodies. And then we could perhaps add DNA methylation as another modality that is missing from our set. So one kind of key limitation why this didn't work right away was because we have the nucleosome in the way. So the methylation binding domain is really poor at recognizing DNA methylation when it's wrapped around the nucleosomes. So we have to actually combine it with the protocols that achieve nucleosome depletion. So we use caltropic salts to remove the nucleosomes while keeping the nuclear envelope intact. So this has been published protocol. We found in a publication from 2017. So if you look at standard nuclei, you can see them kind of individual nuclei intact. After performing this protocol called LAND, we can see that nuclei are a bit less clear because the proteins should be gone, the histones should be gone, but the nuclear envelope is still intact. So we can still use this for single cell analysis. And this caltropic salt is this lithium diiodosalicylate, where also the name LAND for this nucleosome depletion comes. So we tested that, and we have done bulk experiments again, comparing to WGPS, so whole genome bisulfite sequencing data, and then our MBD-TM5 together with LAND, without LAND. I actually see that without LAND, we see a lot of open chromatin M5C, but once we deplete the nucleosomes, we can see a much more even representation and much more reproducible profiles to the whole genome bisulfite. And we can definitely identify both the broad DNA methylation domains, but also individual CpG island methylations. This also works at a single cell level. So we have so far obtained data from cell lines. And when we mix two cell lines, we perform this nucleosome depletion coupled with MBD-TM5 incubation, and then use the 10x Chromium platform. So this should be scalable to tens of thousands of cells. We can find is basically two main populations. This one is split into two clusters, but it basically corresponds to one cell line and this to the other cell line. And we can find very specific DNA methylation patterns in these populations. So this corresponds to bulk WGPS data. So we are really excited about this, and now want to push it further because we can also hopefully what we want to do is combine the barcode this TM5 and combine it with histone modification, so we can do histone mark profiling and DNA methylation profiling in the same cell. Cool. So that was a bit on the technology that what we are doing, but I also want to share a bit about the the biology. So the biologic system that we focus on and we are excited about is a human brain development. And this has for a long time been kind of mysterious process, or like because the tissue is much less accessible than the mouse tissue, um, is much more harder to obtain, and we all only have a specific window so development we can look into. So our window lies in this age between around post-conception week 7 to 13, where we can actually obtain tissue from elective abortions. So we can get the human brains from early gestation. And what we want to do is use our technologies to map the dynamics of the chromatin in these stages, with a focus on the forebrain. So this early fetal development in the forebrain, you can see a coronal section of post-conception week 12 forebrain. So this is very similar to mouse. We have the dorsal part, which contains the excitatory neurons, the proliferatory zones for excitatory neurons, the radial migration of neurons coming outwards from this ventricular zone, and then we have the ventral part, which is proliferatory zone for inhibitory neurons, called also ganglionic eminences, where the cells migrate into the cortex tangentially. So we have been doing single cell ataxic atlas of this has been done before, but as I told you, the ataxic doesn't tell the full story, so we cannot actually. It's difficult to distinguish poised genes, poised cis-regular elements from active, from repressed, and so on. So what we are doing here, Pepe, Jose, and Nichuan, on the wet lab part and on the bioinformatics, they have been collecting samples of the human fetuses, extracting the nuclei, and then running analysis of K20 acetylation and K20 trimethylation as a Markov enhancers and the repressive chromatin. So this is the data we have been able to obtain so far, mixture of multiple different experiments, and we can identify all the main lineages, around 40 different clusters. This is the different time points. So we have started collecting from week 5. So the cells are centered more in the center. They're more progenitor-like. This is corresponding to neuroepithelium of the neural tube, starting to differentiate through radial glia to excitatory and inhibitory lineage of the forebrain. So here's maybe a little bit clearer orientation. So we have the radial glia progenitors here in the middle, excitatory lineage here, and then inhibitory lineage branching, the different identities branching here. So this would be the dorsal part mainly, and this inhibitory part would be mostly the ventral part. So now we're interested, I can show you some markers. So for the progenitors, PAC6 is one of the most prominent transcription factors expressed in progenitors. EMS in the intermediate progenitor cells on the way towards becoming the neurons, and SLC17A7, which is a glutamate transporter, so one of the main transporters for excitatory neurons. On the other side, we have in the inhibitory lineage, we see DLX2 expression and GAT2, which are a transcription factor important for the ventral lineage, and then also for the transporter of or synthesis of the GABA as a main inhibitory neurotransmitter. So we can isolate the dorsal excitatory lineage. We can also perform pseudotype. We find all the usual kind of classical genes being. So this is H3K20 acetylation data, so not gene expression, but we can see kind of similar patterns as on the gene expression level. We see the progenitor genes expressed here early in pseudotype, the intermediate and migrating cells markers in the middle, and then more differentiating neurons genes being K20 acetylated towards the later parts of this pseudotype. So we can visualize this also on the individual gene level, and because it's a multimodal data, we can look at the K20 acetylation and trimethylation at the same time. So for TBR1, important transcription factor for terminal differentiation, we can see gradual decrease of the repressive mark as the progenitors start differentiating, and then increase in the active mark of K20 acetylation. Interesting thing is that we see also genes that you will normally not see in a single cell RNA sequencing, like microRNAs. So we see the microRNA is getting de-repressed and then activated as the cells neurons differentiate, and we know this is a very important proneuronal pro-differentiation microRNA that's expressed in all neurons. So now looking at not at single genes, but hundreds of genes, actually to visualize the dynamics. So we find thousands of enhancers In this case, it's not genes, but enhancers and individual promoters that see a lot of dynamics. So they will acquire K2 cell acetylation at different stages of this differentiation process. And then what is quite interesting is the coordination of the repression, which the activation, the acetylation seems to be much more kind of finely regulated and granular, whereas the methylation is really there. There's a set of progenitor genes that are methylated, they are demethylated as the cells differentiate, and another set which gets more methylated as the cells differentiate. So there is really some, I think there's some fundamental differences between the activator and the repressing system. We can do geoterms correlated with gene expression. So we said also that the expression of the genes that are getting trimethylation is reduced, and the expression of genes that are getting more acetylation is increased. So this makes sense with the known function of these different histone marks, and also the geoterms makes a lot of sense in terms of the neuronal function and so on. There's a little bit data on the ventral side. So now we are in these proliferative zones for inhibitor neurons. There we actually see the three main branches of the ventral. So these are the ganglionic eminences, medial, caudal, and lateral ganglionic eminence. And we can identify really the cell type specific chromatin profiles that has never been done before, and it was really difficult to do using bulk approaches. Now we can really nicely see both the cell type specific profiles, but also the dynamics of this differentiation process. So that's really exciting. So this is really early on, but we are working more and more on this data and trying to explore more, and also collecting more and more samples. So stay tuned for the actual final conclusions from this work. We are complementing our work with the fetal tissue. So as an in vitro model for this differentiation, we are using developing human forebrain organoids. So organoids are derived from induced pluripotent stem cells. We can aggregate the stem cells, create embryonic bodies, and then use small molecules, like in this case is this SB and orthomorphin, to induce neural identity. So this is called neural induction, expand an organoid, and then grow them for a long time. So this is in days. You can grow them up to years, obtain this kind of organoid structures at the end. We have labeling for the PAC6, which I already told you was the transcript factors for the progenitors, and then staining for CTIP2, which is for differentiating neurons. And we are really excited of having this model in the lab now to be able to not only observe the development and describe it, but also to do functional experiments and modulate it on the gene regulatory or chromatin level. And this really also nicely corresponds. This is a staining of the actual fetal tissue. This is the dorsal part, this is the ventral, and you can see the same stainings in the for PAC6 and CTIP2 in the actual fetal tissue. So one of the things what we are doing to also scale this up and be able to profile more nuclei from organoids, we are a little bit repurposing these modality barcodes to create multiplexed nano-CUT&Tag, or we also call it the hash and tag. So we are hashing different organoid batches using barcodes. So we still use two antibodies for two different histone marks, but in a second experiment targeting another set of organoids, we can use two different sets of barcodes and so on and so on. And this way we multiplex around up to 10, seems to be more practical, or samples in the same reaction. And then we overload the chromium with 50,000 to 100,000 nuclei in each reaction. So we can obtain much more data in a single experiment. So this is really, really preliminary, but it's something that Pepe, together with Marie, has been working on, capturing multiple time points, multiple differentiation protocols, multiplexing 12 samples on single 10x ATAG reaction, and they overall be around 36,000 nuclei in this case. We have done more since then, and identify identify different time points of differentiation, and also characterizing the different protocols. So we're really excited about increasing the scale of this kind of technology. And just to show you some markers that it really works, we can see cell type specific genes being acetylated and active in ththese different populations. So we see nano-GIN iPS cells, but then we see a forebrain identity marked by FOXG1, choric plexus, the TTR, and then diencephalon hindbrain. So we can really reconstruct a lot of the complexity of the human brain in the dish, and look at the chromatin profiles of these cell populations, the cell types. Cool. As you also know, for tissue-specific characterizations, the tissue architecture is also very important, and that's why we have been also working together with the lab of Rong Fan and Yangshang Deng at the Yale, and Yangshang is now at UPenn, where he started his own lab to develop a spatial version of the technologies. We use very similar principles. I'm not going to go through that in detail, but we use TM5 transposition either for ataxic or targeted through antibodies and cut-and-tag like protocols. And then we use these microfluidic devices that have been developed in Rong's lab to spatially barcode the tissue. So you can pipette the barcode into these individual wells. This is a 50 wells. Then the microfluidics go from this side to the other side, apply a suction on this other side, a vacuum. So the barcodes got flowed into the nucleus, or into the tissue section, sorry. And then you can do this in X direction, and then on the other side with the Y direction. This is the device, actually how it looks in reality. And then using these oligos that flow, you ligate them to TM5 molecules to introduce these spatial barcodes, and this you can use to reconstruct cell types or tissue-specific with the spatial resolution profiles for K4 trimethylation or K27 trimethylation. And you can identify gene-specific genes that are specifically repressed with a high precision with the spatial coordinates. Yeah, I don't have super much time to go into detail, but this really also tells us about the spatial architecture of this regulation in the tissue. And these are just some examples where we actually did this together with, or Rong's lab did this together with the repressive histone modifications for specific genes, and together with gene expression profiling in the same tissue section. And I think one interesting thing to notice is that actually it's not always that the tissues that don't express the gene. So here's a gene expressed in striatum, and it's the same gene that is actually repressed specifically around the ventricle and in this midline region, and then a little bit in the cortex. But it's not really repressed everywhere where it is not expressed. So I think this the repressive marks also tell a story about which genes are actually actively being repressed through chromatin methylation, and some genes actually just kind of passively repressed through not being expressed and not being actively repressed by the chromatin. So it's quite interesting to look into further. And kind of taking this technology in Yangshang's lab to the next level, what he actually did when he started his lab, developed spatial 5-plex technology where you can look at chromatin accessibility, histone methylation, acetylation, so 2 histone marks, gene expression, and then also protein expression. So going kind of the full circle from the chromatin accessibility, opening, histone mark deposition, transcription, all the way to the protein level. So this is really exciting data. So I think I will wrap up here. Kind of the message I was trying to convey here, that we have this really exciting toolbox for epigenetic analysis, which opens all of possibilities in the chromatin field. So first starting with the first single cell technology to look at one individual histone marks, through multimodal versions of this that we are still working on and developing further in my lab, that we can now use to look at open chromatin and 2 histone marks, DNA methylations coming very soon. And then we also want to expand the number of histone modifications, look at gene expression and other modalities at the same time, that we can really link and correlate these modalities from the same cell, not just from the same sample, but from the same cells. And then also having a spatial version of these technologies that also tells us a story about how these programs are coordinated in space and time as well. And this, we believe that this can be really useful for both identification of and classifying cell types. Of course, you can do a lot of this with transcriptomics as well. And if you are only interested in cell types, you should probably do transcriptomics. But what we can also tell us about the cis-regulatory elements, about the promoters, enhancers, insulators, repressive states, etc. So characterizing these complex chromatin states of regulatory regions. And hopefully we can also integrate this data using computational approaches and inform and gene regulatory networks, as has been done using, for example, multi-omic data with ATAC and gene expression. So we really hope that the histone mark data can also be integrated into these GRM predictions. So with this, I hope I was on time. I will want to acknowledge my lab. So some of this work, or the first part of this work, has been done during my postdoc at Karolinska, where I developed the single-cell cut-and-tag and nano-cut&Tag. And then since starting my lab in 2023, I'm really happy that we can, and we really, I think, on the verge of pushing these technologies one step further, and also applying to really exciting biological questions. So I want to acknowledge all my lab that contribute to these stories, and the protein production facility that really enabled a lot of the TN5 production at Karolinska, and then of course NGI, that sequencing those samples, the tissues that we're getting from Human Developmental Brain Bank, and all the anonymous donors that donate the tissue, and then the lab of my former postdoc supervisor, Gonzalo Castelbranco, and all the funders. And with that, I'm really happy to take questions.